Okay, welcome everyone. I'm Dr. Stephen Linder, and in this lecture, we're going to break up tissues into different sections. We will start with epithelial tissue, then we'll move into connective, muscle, and then nerve. Of the four types of tissues, epithelium and connective are a little bit more challenging. They're not impossible. They're just more categories that fall under epithelium and that fall under connective. Muscle and nerve are a little bit easier in terms of the amount of information that we'll find. Okay, so let's take a look at the four different types. Let's change this to, let's change it to black here. So the four different types of tissues are epithelium, connective, neural tissue, and muscle tissue. And remember, what is it that makes up tissues is that we have to have cells that come together. And when cells come together to have similar structure and function, now we have what we call tissues. And then tissues come together to make organs, and then organs make systems, and then systems make the organism. So epithelium and connective tissue have some major differences. And I just want to point them out here real quickly so that we have a little bit of an understanding. If we were to look at epithelium under a microscope, we are going to see a bunch of cells that are closely packed together. So here are three different cells with different nuclei. I'm going to add another layer to that. So if you notice now we have 6 cells here with a little nucleus in each one, and they're closely packed together. They have very little extracellular matrix, ECM, or extracellular matrix. That is epithelium. But when we look at connective tissue is where we have a bunch of cells that may be far apart from one another. Okay, like this. We have cells scattered far apart from one another with lots of extracellular matrix. I hear a lot of students texting, chiming in, so let me just make sure that everything is fine here. Okay, let's take a look. I'm glad I'm checked in here. Let's go back. Okay, I'm glad. I heard a lot of students texting, so I had a feeling. If there's ever an issue, please unmute your mic and just let me know. But hopefully you can all see this now. Let's make sure everyone can see. Yeah, okay, good. I figured it out. All right, let me just review that real quickly. I know you guys were under a little bit of distress there for a split second. So let me just review what I said. Epithelium and connective tissue. If we look at epithelium, I drew a bunch of cells right here closely packed together, right? 1, 2, 3, 4, 5, 6 cells with nuclei in each one. And there's hardly any spacing between these cells right here. And that's because there's very little extracellular matrix, ECM. In contrast, connective tissue here, I drew 4 cells a little farther apart, and there's more space between them. So we say that there's more room for extracellular matrix. So I'll put ECM up arrow, meaning there's lots of extracellular matrix in connective tissue, and then I'll put a down arrow here, next to the epithelium that there's little extracellular matrix associated with epithelium because they're closely packed together. Okay, so now when we look at the classification of tissues, they're all going to have distinctive structures or patterns or form, right? So they're going to have distinctive structure, and structure determines function, anatomy, and physiology, one of the basic principles. So, um, to perform specific bodily functions, tissues then come together, right? And they're going to organize into organs like the heart, the kidneys, the lungs. And then those organs come together and form systems like the digestive system, the respiratory system, the endocrine system. And then those systems create the organism. All right, now the term histology is the term we use when we study tissues. When we study cells, it's cytology. When we study tissues, it's histology. So epithelium, when we look at epithelium, epithelium is the type of tissue that's going to line and cover things. All right, so when we talk about covering, it's going to cover the external surface of the body. When we think of the external surface of the body, we think of the skin. That's what dermis refers to, the skin. And epi simply means above. So we have a layer of skin above called the epidermis, and then below it would just be dermis. So the epidermis is the external surface of the body, and it's got important functions, but it's considered epithelium tissue. The epithelium tissue also lines the body, and it's going to line the body cavities. You remember those body cavities that we discussed in a previous lecture, the peritoneal cavity, the pleural cavity, the mediastinum, and the pericardium, right? Remember all of those different cavities? So we're going to have epithelium that lines them, as well as tubules. Now, tubules, in the word tubule, we see tube. So anything within the body that has a tube to it is going to be lined here, the inside part of it here and here is going to be epithelial tissue. Now think of where we have tubes in the body. Well, in the throat, the esophagus, the trachea, the windpipe, the intestinal tract, the small intestine, large intestine, those are tubes. The reproductive tract is another tubular system. The cardiovascular system, your blood vessels, your arteries, that's another tubular type of system. So anywhere we have tubes or vessels within the body, it's safe to assume there's going to be epithelium that lines the inside of those tissues. And also glands. Okay, so glands are classified as epithelium. When we look at epithelial tissue, we can name epithelium in a few ways. Number one, there's got to be three questions you're going to ask yourself. When you look at these tissues under a microscope, the first question you're going to ask yourself is, are the cells close together or far apart? Because if you remember, if they're close together, we're going to know that it's epithelium. But if it's farther apart with lots of extracellular matrix, then perhaps we're talking about connective tissue. So that's the first type of question we have to ask ourselves. The second question we ask ourselves is, when we look at it, how many layers do we see? Do we see one layer, or do we see more than one layer? If we see one layer, we refer to that as simple epithelium. And if we see more than one layer, we don't use the word simple, we use the word stratified. So we call it stratified epithelium. And we're looking at the layer that's toward the top, what we call the apical layer, the layer towards the top. That's the second question we would ask ourselves. The third question is, okay, what is the shape of the cells? Are they these flat irregular shaped cells like this. We call it squamous. Are they square or box-like with a nucleus? Then we call it cuboidal. Or are they tall buildings and look like columns? So we would call them columnar. So again, when we name epithelium, we have to ask ourselves three questions. First, we know it's epithelium if the cells are closely packed together. If it's one cell layer, we refer to it as simple. If it is multiple layers, then we refer to it as stratified. And then we ask, what is the shape? It's either going to be squamous, cuboidal, or columnar. So now we look at the pictures here, and this column is simple, and this column is stratified. If we go back to the slide before, we said simple always has one cell layer, as we see in all of these. And stratified is going to be more than one. So this is like 1, 2, 3, 5 cell layers. This is 2 layers. This is 2 layers. So anything more than one is stratified if it's one is simple. Then the second question, and notice how closely packed each of these cells are to one another. Little extracellular matrix. So we know it's epithelium. We know that in this column, it's one cell layers, so it's squamous. It's flat, irregular cells, so it's squamous. And it's one cell layer, so it's simple squamous epithelium. Here, it's one cell layer, and they look like they're cubes, so we call it simple cuboidal epithelium. And then here, they're one cell layer. They're tall columns, so it's simple columnar epithelium. Here, these are flat, irregular, compressed-like looking cells. They're close together, but they're multiple layers, so we call it stratified squamous epithelium. Here, we have the cubes, and they're more than one cell layer, so we call it stratified cuboidal epithelium. And then the last one, these are tall columns. These are stratified squamous, stratified columnar epithelium. Now, one of the things I wanted to point out to you, if you look at the bottom left here, is that these simple columnar look a little bit different than the stratified columnar. One of the things that the simple one has is right here, that little brush border, right? We call that cilia. Cilia. These have cilia, so we could call it simple columnar ciliated epithelium. Now, these cilia are going to beat rhythmically. Let me give you an example. Let's say here is the trachea. Let's say here's the windpipe. Okay, it's a tube, so it's epithelium. And we're breathing in oxygen, so air is going to be coming in here, right? So we'll just put O2 for oxygen coming in, and this is the trachea, the windpipe. Now, let's change the color here. Let's make it blue. So let's do this. So here we're going to have columnar cells, and there's going to be nuclei in each of these, right? And I'm going to make cilia. Let's make this, where's red? Let's put red here. So here, we're going to put these little extensions that are called cilia. And there's many of them, right? And then on the other side also, we're going to have the same thing, right? I mean, these are going to be, they're not cuboidal, these are going to be columnar, okay? I just couldn't make these long enough because of the space in between. But again, similar thing here. and we're going to put cilia here. These are cilia. So these are ciliated columnar epithelium in the trachea. And one of the other cells that's kind of neat that we're going to find here are cells, let's make this green. We're going to call these hidden cells here called goblet cells. These green for goblet. And these goblet cells have a very important function. What they're going to do is they're going to secrete mucus. And the mucus that they release through exocytosis is going to stick to the cilia. So you got a mucus here, and then this goblet cell is going to release mucus through exocytosis as well, and it's going to stick to the cilia here. Now those cilia are going to be beating rhythmically upward. And remember, you're breathing air that's coming in this way with dust particles and debris that can come in and stick to the mucus. But due to the rhythmic contractions of the cilia beating upward, it pushes all the debris up instead of forcing it down, right, into the lungs, into the bronchi, and then eventually into the left lung and the right lung. Now, when people smoke, smoking, however, paralyzes these cilia. They paralyze them. They burn them off and they paralyze them. So if you have no cilia or they're there and just become paralyzed, all the debris that you breathe in and toxins make it into the lungs. No good. But something else happens. If you burn these off, these cells morph into another type of cell. And when cells change from one form into another, there's a term for that. The term is called metaplasia. Metaplasia. And when these columnar cells evolve into or change their shape and morph into something else, they're going to change into squamous cells instead. Squamous cells. Now, we don't want squamous cells in the trachea. We want these columnar cells. So then when one cell type becomes another and they evolve into this through metaplasia, that's precancerous. We call it squamous cell carcinoma, carcinogen, cancer. Okay. Now, I drew these as columnar cells, but the trachea and most of the respiratory tract has something called pseudo-stratified ciliated columnar, right? This one was simple columnar on the bottom left. This one is pseudo-stratified columnar. Now, the term pseudo means false. So false stratified. If you look at the bottom left, notice how all these nuclei are nice and even. It's one cell layer, simple columnar. But here, it looks like one nuclei is at this level, another set of nuclei are at this level, another set of nuclei is at this level, and then other set is at this level. So it gives it the illusion based on the placement of the nuclei that it's stratified, but it's not, right? It's still one cell layer thick from top to bottom. So we call it pseudostratified columnar epithelium. But because of the cilia, we just call it pseudostratified, ciliated, I'll put a C there, ciliated columnar epithelium. Cilia are really important. The goblet cells produce mucus, sticks to the cilia. The cilia are beating rhythmically to trap debris. If people are smoking and there's no cilia, here's what the person sounds like in the morning. We call it smoker's cough. Right? They have to work really hard to force the gunk and the tar and that debris, forcing it from down to up. Okay, so when we look at it from the side view, here is simple squamous epithelium. We're going to have this primarily in the lungs, where the alveoli are. Simple squamous is great for diffusion. where it's going to let certain gases like oxygen to pass through or to allow carbon dioxide to exit. All right, so it's easier for these gases to diffuse through one cell layer than it is to pass through stratified squamous, where we have multiple layers of flat irregular cells. Here on the top in the middle, we got simple cuboidal, down below stratified cuboidal. On the top, we have simple columnar. On the bottom, here's our pseudo stratified columnar, because again, look at the nuclei. They are scattered at multiple levels, giving it the illusion that it's stratified when it's really not. And typically, remember where we have pseudo stratified, we're going to have these cilia that are on top of it. When we have smaller than cilia, we call them microvilli. Microvilli. We have those in the GI tract, in the intestines. Transitional is unique. We find those where we have the urinary bladder, and the urinary bladder can be distended, right? Think of a balloon. If you have a deflated balloon and you draw a square on a deflated balloon, and then you exhale into the balloon, inflating it with air, what happens to the square that you drew on it? It alters in its shape and size. You can't even tell it's a square anymore. Maybe it looks round, okay? So transitional are cells that can vary in their shape and size because of the organ that it's associated with. Just think of the urinary bladder, think of the ureters. They're carrying urine and they can expand. When we look at some of the functions, when we look at simple squamous epithelium, this is what we drew before with oxygen and CO2 passing through. It's really important for diffusion, filtration, and secretion. When we look at stratified squamous, right, that's different than simple squamous. Stratified squamous is many, many layers. We think of the epidermis when it comes to that, like the skin, epidermis. When we think of the epidermis, we think of protection to protect the underlying layers. When we have, oh, we just did this one before, right? Pseudo stratified columnar. This is going to function in the movement of secretion like mucus across the apical surface, because here is the stratified ciliated columnar epithelium, and those goblet cells are going to secrete mucus. So there you go. Simple cuboidal we're going to find around the kidneys, along the tubular system of the kidneys. And within the kidneys, we have a structure called the nephron, which is the functional unit of the kidney. And the nephron has areas that we call the proximal convoluted tubule and a distal convoluted tubule. You'd have to look at a picture of the nephron to actually see these, the proximal convoluted tubule and distal convoluted tubule. But that's where we're going to find simple cuboidal, and they're functioning for absorption and secretion. And that's what the kidneys are doing, right? They're absorbing, they're secreting, and they're filtering. That's what the kidneys do. Transitional, that's the one that's like the balloon based on the urinary bladder. It accommodates distension and relaxation with change in the organ volume. Just think of the urinary bladder, think of the balloon. We drew cilia before. Those are those long, slender extensions that can move secretions and fluids across a cell surface. They can beat rhythmically. The microvilli, we said, are much smaller projections, and they're used to increase absorption. So you're going to find these along the intestinal lumen. So if here's part of the intestine, we think of people who have celiac disease, And let's say they have these columnar cells as well in the intestines. They should be a little bit longer. Let's see if I can draw them like this a little bit better. Again, nuclei in each. But there's going to be microvilli, these small microvilli on the top of them, and that's for the absorption of vitamins and minerals and water, and all the micronutrients can go through there into systemic circulation. And when people are gluten sensitive or they consume gluten and their body breaks down these microvilli, then there's lots of malabsorption diseases that take place because you can't properly absorb the nutrients in the food. Okay, that's an important concept. So again, the epithelium, to kind of review, right? Once we establish the cells are closely packed together, they could be either simple epithelium or stratified, depending on one or multiple layers. They're either irregular flat-shaped cells, they're cuboidal cells, or they are columnar cells. And the squamous is for primarily diffusion and protection and secretion. The cuboidal cells, we find them along the proximal and distal convoluted tubule of the kidneys. These are for secretion and absorption as well. And then the columnar, we'll see these in the respiratory tract around the trachea. We'll see it along the digestive tract as well for absorption of nutrients. The pseudo stratified, we said that the nuclei lie at different levels, so it gives it this false appearance of being stratified, and they're often associated with cilia. That was the example of the trachea that we provided earlier. Transitional epithelium, this is a type of stratified squamous, but the cells, depending on the organ, whether it's distended or not, those cells start to stretch. So the shape of those cells start to vary and change. We'll find it especially in the urinary system, such as the urinary bladder, the ureters, and the urethra. So they could be flat. When the organ is full, they could be rounded when the organ is empty. Okay, just like a balloon. If you draw a square when it's empty, it's a square, and then when you inflate it, it alters its shape. So let's take a look at this picture, just see where we find some of these cells. Let's take a look at the top left. So when we see simple squamous epithelium, these are found in the alveoli of the lungs, or the air sacs of the lungs. And this is going to allow oxygen to come in, or CO2 to diffuse out, right? It does it nicely when it's one cell layer thick. In the right over here, here's where we have columnar cells, but the nuclei are scattered at different levels, giving the appearance that it's stratified, but it's not. So it's pseudo-stratified ciliated columnar epithelium. We find it in the respiratory tract. We said smoking can paralyze or damage those cilia. Simple cuboidal epithelium, those are found in the kidney, especially the proximal convoluted tubules and the distal convoluted tubules of the nephron, which is associated with the kidney. Simple columnar, these are found in the GI tract or the intestines. And again, you can also have not cilia here, but you can have these microvilli, much, much smaller microscopic filaments for the absorption of nutrients. And then stratified squamous epithelium, right? It's the top few layers. You always look at the apical layer towards the top. We're not looking at the bottom layers. We're looking at the top portion. So it's definitely flat, irregular cells, multiple layer, stratified squamous, not only the esophagus, but even the skin, the epidermis. Where here, these were cube-shaped, also one cell layer thick. Okay. So epithelium could be simple squamous, simple cuboidal, simple columnar. That's on the simple side. And then on the stratified side, we can have stratified squamous, stratified cuboidal, stratified columnar, and the two fun ones, right? Pseudostratified, ciliated columnar epithelium or transitional epithelium. Good place to take a break. We'll see if anyone has any questions. Okay, everyone doing good? Any questions? Anyone have any questions before we go on the epithelium? We're all good? We'll move on. Awesome. Thumbs up. Good, good, good. Great. Okay. All right. We're going to move on to connective tissue. Here we go. Let me turn my camera off here. Let's get the screen share on this time. That was the mistake last time. There we go. Your connection is poor. Why is my connection poor? Let me say, it shouldn't be poor. Nope, I got a good connection. Okay. Let me get this one out of here. Here we go.